facilitating this evening's presentation as well as the Q&A section we will have after the lecture. To start, just a few quick notes. Our session will start with a talk by Dr. Shankle about treating and preventing neurodegenerative diseases. In the final 15 minutes, we will open things up to the Q&A. If you have questions you'd like Dr. Shankle to address, please use the Q&A function in Zoom. You should have a button at the bottom of your screen you can click on and type a question that will come to me. I will then bring those up during the Q&A section after the lecture. I may not get to all of them, but we will try to get to as many as we can, and if there's anything that isn't answered, we can also try to get that answered after the lecture. Video and audio are disabled for attendees, so if you are shouting or waving at the computer, have no fear, know that we cannot hear you. If you have comments or questions, you can message me or use the Q&A feature. Lastly, we are going to post a recording of tonight's webinar on YouTube and on the Shankle Clinic website in the coming weeks, so stay tuned for that. And if you know anyone who wanted to attend tonight but couldn't, we will send the recording to them. Okay, so that's it for administrative items. So to start, I'll give a brief introduction of our host, Dr. William Shankle. Dr. Shankle is one of the country's leading experts in Alzheimer's and other age-related neurodegenerative conditions. He spent over 3 decades treating patients fighting these diseases, conducting research to advance our knowledge of the brain, and developing new treatments to delay or prevent the progression of cognitive impairment. In 1997, Dr. Shankle founded the Shankle Clinic, a neurology practice headquartered in Newport Beach, California, which is focused on the management and treatment of neurodegenerative conditions. Prior to founding the Shankle Clinic, Dr. Shankle served as the clinical director for the Alzheimer's Research Center at UCI, which he co-founded. He also served as the director of Memory and Cognitive Disorders Program at Hoag Neurosciences Institute. Currently, in addition to leading the Shankle Clinic, Dr. Shankle serves as the Chief Medical Officer for MBIT Corporation. He is the Voltmer Endowed Chair at Hoag Memorial Hospital, and he's a clinical PI for a 5-year NIH grant at UCI. So without further ado, I will go ahead and pass the reins to Dr. Shankle. Thanks everyone for joining.
Thank you, Christopher. That was a very nice introduction. I'd like to tell you about what is the cause of...
Will you share your screen, Dr. Shankle, so we can see the lecture? Thank you.
Okay. I'd like to tell you about how we can prevent Alzheimer's and other degenerative brain diseases, something that is not anymore in the future, but is now a real possibility. And it isn't that difficult to do. I say it's a numbers game, and you will see what I mean. Okay, so a lot of people get diagnosed with MCI, mild cognitive impairment, or dementia. And the first thing that you should know is that that isn't a diagnosis. Just like if a car doesn't work, if it doesn't start, it might be due to many things like a motor failure, a dead battery, a sensor failure. So the car not starting isn't the diagnosis, it isn't the cause. Similarly, dementia is simply a measure of severity. It's not a diagnosis of the cause. And just like anything in medicine, once you identify the cause, then treatment becomes much more effective. Mild cognitive impairment is also a popular term, also called MCI. It is not a diagnosis. And if you're told that it's a diagnosis, find someone else. What causes Alzheimer's as well as other degenerative diseases? Simply put, stress on cells. And we'll talk a little bit about what sources of stress on cells occur, but that is simply the most well-supported cause of degeneration in the brain as well as the body. So we call this the cell stress hypothesis of neurodegenerative disease. And this is a picture of a cell, and the different colors represent different parts of the cell. And each part is made up of thousands of proteins. Uh, each protein has a particular job and a particular shape that it gets folded into in order to perform its job. Typically, a cell will have more than 100,000 different proteins. Now, in order for a protein to work inside a cell, it has to be folded into a particular shape. And this basically is a picture of how this folding can occur in a series of conversions where it then gets sent back to the cell somewhere where it belongs, like if it works in the mitochondria, right here, this red looks like a worm, kind of. It will get sent there to do its job. Now, what sources of cell stress are there? I've outlined the big ones. Genes can produce an increase in protein misfolding, which it causes cells, which is a form of cell stress. Medical conditions, chronic medical conditions, if they're not well managed, psychiatric conditions, if not well managed, such as depression, bipolar, anxiety disorder, post-traumatic stress disorder, cancer, as well as cancer treatment, can cause stress on cells. Recent infections, such as urinary tract infections or acute illnesses, such as COVID, will cause stress on cells. General anesthesia from a surgery. And then treatments themselves can have adverse effects that stress cells. Injury to the body or brain, environmental toxins and hazards, and nutritional deficiencies, as well as inactivity physically or mentally. All of these things can actually damage proteins in cells. So we have to have a way of repairing that damage. And so what the cell does when a protein is damaged, it makes a new one to replace it. This shows the series of steps that the cell goes through to make a new protein. And then, so it starts by the nucleus synthesizing a new protein, and then it gets folded, and then it gets transported to its proper location where it functions. This whole process is a lifelong process, and the scientific term to preserve cell function is called homeostasis. So again, stress can be from many, many different sources: chronic diseases, lifestyle, heredity, trauma, acute illnesses, and psychiatric conditions. Now, this is actually what misfolded proteins look like. I borrowed this slide from Dr. Charles Glabe at UC Irvine. Protein folding and misaggregation and aggregation occur when the cell stress exceeds the capacity of the cell to replace the damaged proteins. And when this occurs, then errors are made in the folding process. Those errors in folding lead to different misfolded proteins aggregating together to form these strange and bizarre looking three-dimensional shapes. They have a abbreviation called CAMPS, which stands for cytotoxic or cell death, aggregated, misfolded protein structures. And these aggregates, they attach to the cell membrane, which is shown at the bottom of the slide. You can see the aggregate that's sticking into the cell membrane. And the way a cell membrane functions is that it communicates within the cell membrane laterally. So when it runs into one of these aggregates, it's like a roadblock, and the cell starts to not function properly because the membrane is essentially where all the action occurs in terms of a cell's function. Now, you have a number of defense systems to get rid of these misfolded proteins if they do develop. So the most important one is your immune system. Your immune system, when these misfolded aggregates develop, your immune system makes antibodies, and those antibodies specifically target different shaped misfolded proteins in your brain or body. And it makes those antibodies and it sends them to find these misfolded structures inside cells. And then you can see these gray and kind of greenish structures with AB. That stands for antibody. And so there are many antibodies that will bind to this misfolded protein, which has got a amyloid core, and then it's got a bunch of purple structures sticking out that the antibodies recognize. Once that occurs, there's something called a chaperone. Just like when your daughter went on a date, she needed a chaperone. Well, the cells have chaperones too for the antibodies and these misfolded proteins, and they escort them out of the cell. They're then channeled through spaces in the brain right to the center of the brain where there's a huge vein, and it literally dumps these aggregates of the antibodies and the misfolded proteins into the vein, and it leaves the body, the brain, and then leaves the body. Now, an important discovery that is, in fact, it's extremely important, is that Charles Glabe at UC Irvine, he addressed a really difficult problem. You may have heard of the term biomarkers. These are measures of a disease. Now, these biomarkers are attempting to measure these different misfolded protein shapes or different CAMPS structures. And the levels in the blood and the spinal fluid are extremely low. If we wanted to develop a blood assay, you have to have extremely sophisticated technology to detect the levels of these misfolded proteins in the blood. And some of them, which are very important, such as in Lewy body disease and frontal temporal disease and chronic traumatic encephalopathy, some of these levels are so low that they cannot be detected with current technology in the blood. So what Dr. Glabe said is, well, instead of measuring the misfolded proteins, why don't we measure the antibodies against them? And lo and behold, there were tons of those antibodies in the blood. And what you see here on the left-hand side are 22 rows, and each of them has a set of letters. Some of them are highlighted in red. Each of those rows represents a different antibody that recognizes a different misfolded protein. And this is in a single patient with Alzheimer's. So the notion that all these misfolded structures are all the same and that a single antibody can recognize them all, it's simply not supported by what Dr. Glabe has shown. And this has been replicated many, many times. The majority of people have anywhere from 20 to 100 different antibodies against different weird shaped misfolded structures in the brain that are the cause of degenerative brain disease. So this is one reason, I think at least, why you've probably heard that there were 3 drugs approved for Alzheimer's. Each of these drugs bind is a single antibody. It's not 22, it's only a single antibody, and it will only recognize some of the misfolded protein structures in a person's brain who has Alzheimer's. And this could explain why the treatment effect is not that big. It's measurable, but it's not a huge benefit, a huge treatment effect. Um, the other problem, which I'll just mention because I know there's a lot of interest in these treatments, is that you have to be 100% sure that this person who gets these antibodies that have been approved by the FDA, so-called lecanemab, donanemab, aducanumab, you have to be sure that the person doesn't have leaky blood vessels. This is extremely common as a co-disease with Alzheimer's in persons who have so-called ApoE4 gene. That gene is responsible for tripling the rate of production of one of the misfolded proteins, amyloid. And that particular one tends to accumulate not only in the brain, but in blood vessels. And when they attach in blood vessels, they punch holes into the blood vessel wall and make it leaky. You do not want to give one of these new FDA-approved drugs in a person with these leaky blood vessels because they greatly increase the risk of a brain hemorrhage, which can be fatal or severely disabling. So in summary, the cell stress hypothesis of degenerative brain disease says that uncompensated cell stress leads to increased protein misfolding, aggregation, and cell function, cell dysfunction, and if not removed, degenerative disease begins. Now, in terms of treatment, what I've learned in the last 10 to 15 years is that if we can get a measure of the disease in the brain, then we have a way to measure the effect of any treatment, regardless of what it is. And this graph simply shows what I call a numbers game. Let's say we start by getting a level of the misfolded proteins at baseline, and then we start our treatment. And then after a couple of years, 1 to 2 years, we repeat that measure. And if the level of these misfolded proteins has gone up, almost 100% of the time, a person will have declined. If the levels of the misfolded proteins haven't changed at all, as shown by the yellow arrow, people almost always remain stable. However, if we have been successful, the treatment has successfully lowered those overall misfolded proteins, we do see partial improvement. And I see this virtually every day in at least some of the patients that I take care of. So if we turn our focus now, how do we most effectively prevent or treat Alzheimer's or any other degenerative disease? Well, I strongly believe in measurement, and that's why I call this a numbers game. So the first step is you want to detect the underlying disease as early as possible, so there's as little damage as possible. If you detect it before symptoms develop, then prevention is possible, prevention of the symptoms. If symptoms have developed, then treatment will be most effective when started early. Now, one of the exciting things, I've actually been involved with this for more than 20 years, is that a form of artificial intelligence has been used by my colleagues to model the underlying processes in the brain that are involved when a person takes a memory test. And it took us 24 years to make this discovery, but now we can accurately predict whether a completely normal person will develop Alzheimer's within 3 years. So we can predict whether a person will become impaired, and we have a 3-year lead. That means that we can then proceed with a diagnosis to identify the underlying disease, treat it with the hope and probability that symptoms will never develop. I think that's a major step forward because this is a simple 10-minute memory test that can be done, and it tells us what we need to know to take the next step. The second step is to diagnose the causes. Effectively treating any disease, no matter what, you have to diagnose the cause. And in the brain, we do brain imaging called quantitative MRI. We do blood tests to identify other sources of cell stress that cause cognitive impairment. We also have biomarkers that measure these misfolded protein levels. We have cognitive and functional tests that measure the function of the brain at a higher level than these misfolded proteins. And we also take a history to identify the risk factors for cell stress to make sure that they are being well controlled. And just to reiterate, any chronic disease, if not well managed, will increase cell stress and accelerate the development of degenerative disease. So this just kind of is a picture to illustrate how we approach the diagnosis. It's more reiteration of what I've already said. We have the biomarkers that we can use, both spinal fluid and hopefully very soon blood. I don't think they're entirely there yet. We have quantitative MRI measures. We have cognitive and functional tests, and we can measure the underlying processes with artificial intelligence models. This is a summary of approximately, I think it was 550 patients in the Shankle Clinic who underwent a spinal fluid test, and we measured the levels of some of their misfolded proteins. And what you see is that the graph is divided up into quadrants. In the middle quadrant, the narrow column at the bottom below the red horizontal line, that group only has pure AD. They don't have other diseases. Now, everybody below the horizontal red line has met the most sensitive criteria for Alzheimer's disease. And what this is telling us is that the majority of people with a diagnosis of Alzheimer's disease who have the pathology, only 10% of them have just AD. 90% have Alzheimer's plus some other disorder. Could be degenerative, it could be cumulative cell stress factors that are easily treatable. But 90% of people with Alzheimer's have more than Alzheimer's. So if you're going to effectively treat, quote, Alzheimer's, it's only going to work 10% of the time. You have to identify as many of the causes as you can to get the most effective treatment. So finally, we treat the causes and we quantify the change. That's the numbers game. If a treatment is effective, it must measurably reduce the cause of the disease. And I'm just going to show you one example. Like I said, every day I see the results of this type of an analysis in patients who do show real improvement over time, even though they have a degenerative brain disease. This is an example of a 68-year-old gentleman who had mild memory loss. The evaluation showed he had Alzheimer's plus brain hemorrhage plus more tangles, another misfolded protein that's found in Alzheimer's, but it's also found in other conditions, more tangles than we just be expected by Alzheimer's alone. So this person had 3 different diseases of the brain. The left-hand side with the colored bars of red, blue, and orange are measures of 3 of the misfolded proteins, and they measure the amount of change per year from the first biomarker test to the second over about a year. And each of these biomarkers or causes of the disease decreased by more than 15% per year, every one of them. And if you accept the standard dogma, these biomarkers are supposed to increase by anywhere from 5 to 10% per year. So they're going in the opposite direction. On the right-hand side, you'll see another bar graph, gray and blue and yellow and green. And this is a measure from the picture of the brain, the quantitative MRI, representing the changes in brain volumes. And aside from the blue bar, the other bars are showing a rate of change of less than 1.5% per year in brain volumes for the majority of the brain, the cortex, one of the key memory areas called the hippocampus, and one of the first areas affected by Alzheimer's, the perihippocampal occupancy. All of these are changing at a rate of normal aging. Okay, even though this person has Alzheimer's at a very early age, which tends to progress faster. So this is simply an example of how we can show that you can actually treat the disease. And if we then look at the function, this is an example of a brain function scale that's very reliable. And what it shows is that this person who has a mild memory loss has been in the same stage for 5 years, where the average duration without treatment is 2 years. And deviation from that is 1 year. So this person has stayed stable for at least 2 years longer than the untreated average state. So effectively treating the cause of the disease improves all of the underlying parts because they have to work together. And the underlying parts are the cognitive function, the functional capacity of the person's brain, the brain physiology, the anatomy. We can measure all of that. And then because they all are connected to each other and work together and compensate together, that allows us to interpret the effect of a treatment. If they're all pointing in the right direction, we can reliably interpret the effect of the treatment. And I find this is true at least 95% of the time. It's very reliable. So I think there's a very clear way to, first of all, identify the cause, measure how much of the causes are present, initiate a treatment, and then measure the effect of the treatment on the entire brain system. So going back to that numbers game, I've now treated more than 300 patients for up to 10 years who have, quote, incurable degenerative brain diseases. And they have either stopped progressing, partially improved, or substantially delayed their progression. So this is something I've come to expect. I don't expect decline anymore. I expect stabilization and partial improvement. And when I do see decline, I know that means the treatment has to be adjusted. So what's next? Well, for a long time, I've been working with Dr. Glabe, and he and I have now gotten approval from an institutional review board to do a blood test that takes a small amount of blood from a person and can identify all the antibodies they have against their brain disease. The antibodies in this person are shown by the different colored circles. And this is a very simple and rapid way to identify the amount, the different types of disease that a person has in their brain. Once we have accomplished that phase, which is to take a sample of blood, identify the underlying disease, as well as the antibodies that are removing that disease, the next phase will be to extract each person's antibodies specific to the brain disease and then have them administer them by inhaling them through the nose. And the reason for doing that, inhaling it through the nose, is that there's a barrier between the blood and the brain. And if you give antibodies by intravenous infusion, injecting them into the blood, only 0.1% of those antibodies actually get in the brain. That means 99.9% don't ever get into the brain. And yet, for more than 50 years, the standard treatment with antibodies has been to infuse them into the blood. This simply makes no sense for degenerative brain diseases. 10 years ago or more, I identified a study that showed in the mouse that when they inhaled their antibodies, they got into the brain up to 1,700 times better than when they were injected into the blood vessels. And that's when I started to use the intranasal delivery. And the beauty of it was several-fold. One, it reduced the cost of a monthly treatment from when you give it intravenously. The average cost was $10,000 a month. It's reduced that cost to about $400 to $600 a month. Secondly, when you inject these antibodies into the blood vessels, they can bind to the amyloid in the blood vessels and cause brain hemorrhage. To date, I have seen 0 cases of brain hemorrhage by using intranasal delivery of the antibodies one can generally prescribe. Third is that even though the doses are anywhere from 30 to 50 times lower when administered intranasally compared to intravenously, the treatment effects appear to be as large or larger than when one gives the same treatment intravenously. So it doesn't make a whole lot of sense to me to try to, use intravenous, and yet this is the only way that the currently approved FDA treatments are allowed to be administered, is intravenously. I've tried to get them to do a head-to-head trial using their drug with intranasal versus intravenous, and 2 of the 3 companies have flatly refused to do so. Now, the final thing is that once we have accomplished the second phase, which is extracting a person's antibodies against their brain disease, then inhaling those antibodies will remove 20 to 100 times as much degenerative brain disease as we can currently accomplish by the prescribed antibodies that are derived from thousands and thousands of blood donors. So we have the study approved. We've developed the budget for it. The first phase, in order to do this for all Shankle Clinic patients will cost about $1 million. That's not that expensive, given that a clinical trial costs several $100 million. We've also established a nonprofit foundation, the Foundation of Memory, in order to raise the money, and we'll also be writing grants to accelerate the initiation of this trial. I hope this has been helpful in some way, and I'll be happy to answer any questions.
Thank you very much, Dr. Shankle. I have one question here, which is, what is the role of lifestyle changes in treating, these diseases and disorders?
The role of lifestyle, that's very interesting. There are a number of, can you still see me, by the way?
Yes, yeah.
Okay. So there's a number of studies that have been done internationally looking at the effect of lifestyle on disease progression in Alzheimer's, in the mild stages and the mild dementia stages. And what they did was they looked at nutrition, cognitive or mental stimulation. They looked at managing a person's medical conditions, and they also looked at physical exercise. And they found consistently a very beneficial effect in delaying the progression as well as improving cognition. So we started a study, a 2-year study at the Shankle Clinic several years ago, and we published it last year. And it showed, this study was called the COCO study. We used the same lifestyle recommendations that have been recommended internationally. And we had 2 groups. One group got the recommendations. The other group also got the recommendations for lifestyle, but they also got a life coach. A life coach would help kind of tailor the recommendations to the individual. And then periodically they would check in, the patient would check in with the life coach to help ensure compliance. And that was the big factor, is compliance. People have a lot more trouble maintaining a consistent lifestyle interventions on their own than when they have somebody that they have to report to. Everybody's a little different. Some people have no trouble. Other people can't do it without a life coach. And so what I've learned is that the treatment effect of our study, the COCO study, was larger than all 3 of the newly approved Alzheimer's drugs. People did not decline for over 2 years in those who had a life coach and maintained the lifestyle interventions. So I think it's a very significant effect, and it's so significant, I was so impressed that I'm actually using the life coach for myself.
Well, thank you, Dr. Shankle, for that answer. We have another question here. What are the effects of this treatment on Lewy body disease? And I think in general, what are there's a lot of talk of Alzheimer's. Does, intranasal IgG hit all neurodegenerative conditions? How does it work?
Okay, so this is regards to Lewy body disease as well. That's a member of a family of diseases that have a particular misfolded protein structure called alpha-synuclein. And alpha-synuclein is just another weird shaped misfolded protein. It happens to be in a particular part of the neuron. There are treatments that can block its production and remove it. One of them is green tea extract. Another one is Percepta, which is a combination of the cat's claw plant and oolong tea extract. the intranasal IgG can lower the alpha-synuclein misfolded proteins. There may be, I can't think of, I, no, I can't think of another. So there are at least 3 treatments that can do that, lower alpha-synuclein. So it's not as well researched as Alzheimer's disease, but I have treated quite a few people with Lewy body disease, and their quality of life has been enormously better, stabilizing for years, sometimes showing improvement. Another synuclein disorder is Parkinson's disease. And again, these are diseases that tend to affect the brainstem. And one of the exciting things is that if you inhale the antibodies through the nose or whatever drug it is, it gets into the brainstem 1,700 times better than if you injected it into the bloodstream. And it kind of made sense because a lot of the time, the people who showed the biggest treatment response had either Lewy body or Parkinson's disease or another disorder of the brainstem. So I think that, yeah, there's no, if you go to the Lewy body Society website, they'll state there's no effective treatment. I don't think that's true. And I just, again, think that degenerative disease is a numbers game. And if you can identify the pathology, you can treat it and measure it.
Thank you, Dr. Shankle. That does, raise some interesting questions about, why is there so much skepticism about some of these other treatments, using supplements to treat neurodegenerative conditions and whatnot?
Yeah, it's an excellent question. And some of my colleagues have raised arguments about the doubting the efficacy of it. One argument is that the quality control of the supplements is poor. And while that may be true for some, there is something called GNP, which has to meet government requirements for natural products. And so if they have a GNP label or stamp on the supplement, that tells you there's a guaranteed purity that has to be met. So that's one way to be sure about a supplement. And another is that there is a strong, unfortunately, a disincentive for researching supplements. There's a huge profit margin motive for developing drugs and selling them. So you simply don't see as much research. And but if you comb the literature, you will find studies that very specifically can identify this supplement will lower tau. This supplement lowers alpha-synuclein. This one lowers beta amyloid. That's actually why when I do measure the misfolded proteins, I need to know which ones are abnormal. That helps me select the best supplements and antibodies and medications.
Thank you, Dr. Shankle. We have another question here that's asking, is there a genetic test for Pick's disease? Are there any drugs to slow it down?
That's a really good question. So Pick's disease is a form of frontal temporal lobe disease. It has a different set of misfolded proteins. And there is a gene panel that you can get with a blood test or saliva test that looks at all of the genes that are associated with different frontal temporal lobe disorders. And when I have a patient that I suspect has a frontal temporal lobe disorder like Pick's, which is really exemplified by the story of a famous neurosurgeon um, who one day decided to hitchhike across the United States. I just, that characterizes Pick's disease. It's very bizarre behavior. It's usually socially inappropriate. A lot of times there's sexual disinhibition, and it just really ravages the behavior aspects of the frontal lobe. So there is a gene panel, and there are identified misfolded proteins. I have only had a chance to treat about 2 patients with frontal temporal lobe disease, and I can't say that I've seen a very significant effect. And I kept asking myself, well, why could that be? I think one reason may be is that if you take thousands of donors of blood and you pool their blood and you make the antibodies and you sell it and we prescribe it and they intranasally inhale it, the proportion of patients who have frontal temporal lobe disease is extremely small. So not many of people will be contributing antibodies against frontal temporal lobe disease. I'm hopeful that the analysis that we do with the Dr. Glabe's blood samples and then extracting each patient's antibodies against their disease will effectively treat frontal temporal lobe disease as well as diseases like ALS and some of the progressive supranuclear palsy, stiff person syndrome like Celine Dion.
Thanks, Dr. Shankle, for that answer. We have another question here that's asking about lion's mane. And more specifically, they're interested, is it only used to treat dementia or can it be used to prevent dementia as well? And then if so, they're also wondering if you know if lion's mane is better than Percepta or if that might be sort of, patient specific.
Yeah, it's more disease specific. So, for example, what does lion's mane do? Well, lion's mane is very similar to estrogen. Estrogen increases a growth factor in the brain called nerve growth factor. And that growth factor helps preserve the nerve cells that release a particular transmitter called acetylcholine, which is very important in visual abilities, in executive function, in focus, concentration, multitasking. So in one application of it is in women who cannot take estrogen, they will get low levels of nerve growth factor and they will have loss of these neurons that release this transmitter. You can give lion's mane at a dose of 2 to 3 grams a day, and that can maintain the nerve growth factor in women who can't take estrogen for whatever reason. And then the other group is that in Lewy body disease, more so than Alzheimer's, the greatest deficit they have in their brain is the loss of neurons that release this acetylcholine transmitter. So giving them lion's mane helps preserve those neurons, and I try to give it as early as possible to maintain that population of nerve cells.
Thank you, Dr. Schenkel. We have another question here about, what are some of the nutritional diets that ward off the onset of dementia, and how important is it to, focus on diet, whether you have a condition or, you're predisposed genetically for one?
I suspect there's a lot of varied literature out there. The one that has most consistently shown a significant risk reduction is one that I think most people are familiar with, called the Mediterranean diet. It tends to be low in red meat and dairy products. It tends to be high in roots, vegetables, chicken, fish, beans, those types of foods. And interestingly, the three city study in France was a longitudinal epidemiological study that looked at the components of the Mediterranean diet that reduced the risk of Alzheimer's and dementia. And they found that the most significant effect came from olive oil.
Wow, fascinating. Very
Yeah.
fascinating. Um, so we have a number of other questions coming in. I just want to let everyone know that if we don't get to your question today, I will save all of the questions and we'll have the doctor answer them offline and we can, send them around just in summary form, um, after with the recording of this video. So the next question that we have, because we do still have a little bit of time, does a patient need to have CSF amyloid beta analysis in order to undergo treatment with INIG, which is intranasal IgG?
Um, the answer is yes. And let me explain. Uh, Very recently, there were very exciting studies that showed that blood tests could replace the spinal fluid test to measure these misfolded proteins. And they were very credible journals. So we, followed suit and we started to order blood tests to replace the spinal fluid test. We compared the spinal fluid test to the blood tests in a number of different patients, and we found a tremendous inconsistency of the blood tests. They were simply not reliable in measuring the disease. So at present, I'm still using spinal fluid. Uh, I will obviously switch once we can get reliable measures of not only the amount of disease, but the change as a response to treatment. That's equally important. And literally speaking, there's less than one, there actually is either one or no studies on how these measures change, how reliable the blood tests are in terms of measuring change. So we have no idea for that. That's why I continue to use the spinal fluid biomarkers. And there's an additional reason that's very important. Um, the additional reason is that I can also measure the integrity of the barrier between the blood vessels and the brain. We now know that if that barrier breaks down, that is usually a harbinger of much more rapid progression of the disease. And so being able to measure whether that barrier is still intact is extremely useful. And I just today saw a patient whose blood-brain barrier was 3 times as leaky as normal. And that was several years ago. I had put her on intranasal IgG, and 2 years later, her barrier went from 3 times leaky to just barely above normal. It was like a twofold reduction in the leakiness of the blood-brain barrier. I was absolutely delighted to see that because that's a big factor in treatment.
Thank you, Dr. Shankle. Do you think those blood biomarkers will be coming in the next couple of years? Is that your hope?
Yeah, my hope is that they will be coming in the next couple of years. The technologies, the level, the real problem is the amount of misfolded protein in the spinal fluid is much higher than in the blood. So they need totally different technology to detect the levels in the blood and then to detect them accurately so you can measure change is another challenge. But I think, the technologies are developing. Um, and don't forget that it may be that Dr. Glabe's approach is even better than that, because there's tons of antibodies in the blood that we can detect.
Thank you, Dr. Shankle. I think we maybe have time for two more questions. So our next question is the process of proteins becoming misfolded a statistical process? And does it happen across different ages? You know,
So is the process,
is it linear? Is it exponential? And how does it
oh boy,
you
okay.
get the same? I don't know.
So,
I'm just adding to that question. I'm not the math guy here.
well, that's a good question. And I think I'm not sure I can answer that accurately, but I can say that there was a beautiful study. It was a computational study that modeled the aggregation of amyloid into different shaped misfolded proteins. And what they showed was that there are many, many different pathways of misfolding that amyloid can follow to create different shapes. And it seemed to be a statistical process, or at least somewhat random, in the sense that these aggregates are inside cells, they're floating, and so they will happen to contact another aggregate in different ways. Almost, you could almost model it randomly. So I think there's a lot of truth to the process having a statistical nature.
Hmm. Thank you. Very interesting. Our next question
Hey,
is
just a little comment. These questions are better than the ones that the university professors ask.
well, it's a smart patient and caregiver population. The next question, what is your opinion regarding hormone replacement therapy in regards to preventing Alzheimer's disease? And I just want to sort of, separate this out. What is your, how do you feel about like Lupron, castration therapies for, folks who have had prostate cancer, and also, estrogen for folks who have gone through menopause? Um,
There's two sides. The question can be divided into two parts. One is the hormone replacement therapy. How does it impact the risk for developing Alzheimer's? And that would be with regards to postmenopausal women. Um, there was a study many years ago called the WHIMS study, Women's Healthcare Initiative. I think it was for menopause state. Anyway, they finished their study and they concluded definitively that taking estrogen caused Alzheimer's. And for several years, this was dogmatically stated. Well, it turns out that the women that participated were over 65 years old and had had menopause or had gone through menopause more than 20 years earlier. What the subsequent research showed was that if a woman supplements with estrogen within 5 years of menopause, their risk for developing Alzheimer's is lower. Okay. So the real problem was these women who participated in the WHIM study simply didn't have the opportunity to take estrogen within 5 years of their menopause. So that's one issue. We also published a study many, many years ago. This was called the National Mortality Follow-Back Survey of 100,000 women. And this was in the era when hysterectomies were performed for various reasons, cancer, et cetera. And most women were not put on estrogen. So you had an example where you induce menopause with surgery, and then you don't treat them with estrogen. It turns out that women who had a hysterectomy during this time were more than twice as likely to become demented. So that was consistent with the idea that it is useful to supplement with estrogen within some critical window that's within 5 years of menopause. So that's one side of the question. The other side of the question is for men. And, men tend to not get, they kind of get ignored sometimes when it comes to studies. We don't see too many andropause or menopause studies, as I like to say. So, but I've become fascinated with prostate cancer because what happens in prostate cancer is that men who develop recurrence after their initial treatment get put on what's called androgen suppression therapy or androgen deprivation therapy. They literally turn their testosterone levels down to zero. And there are now a number of studies that show that when that happens, that these men within 5 to 10 years develop Alzheimer's. So when you cut out testosterone, it turns out that testosterone, it removes amyloid in two different ways biochemically. So you take that away and it allows the aggregation of amyloid, two different ways to aggregate amyloid more. And I now have a number of patients with prostate cancer who've had testosterone suppression and have developed Alzheimer's disease within 5 to 10 years.
Okay, well, thank you, Dr. Shankle. Any concluding thoughts?
It's been a delight. You're a good host, better than David Frost and Charlie Rose. Thank you
I
very
don't think
much,
so.
and I appreciate all the families who take the time to learn more. And I'm very positive that before I pass on to better pastures, we will have prevention.
All right, well, thank you so much to everyone who joined tonight. And if one of your questions was not answered, I have saved questions, so I will make sure that the doctor answers them and we can send those around after. So thanks, everyone. Have a great rest of your week, and we will have one more webinar before the end of the year. Take care.
Bye.